All right, guys, so here's DNA. From what you already know, what's the shape of DNA? It's a double helix. Now what that means, it's two strands of chemicals. It's sort of like a ladder that's been twisted. It's like a three-dimensional twisted ladder or spiral staircase. Now, for the whole semester, we've been talking about four groups of biological macromolecules. We've been talking about lipids, nucleic acids. We've been talking about proteins, and what's the one I haven't gotten up here yet? Carbohydrates. All organic macromolecules. Which one of those four is DNA a member of? Nucleic acids. Nucleic because DNA is usually found where? In the nucleus. And it turns out it's acidic because of this phosphate group. Phosphate groups give off what? What do acids give off? H pluses, hydrogen ions. Acids make things, the pH drop. Phosphate groups are acids. Other than DNA, what's some other examples of nucleic acids that we've already dealt with in this class? Well, he said RNA, obviously. But things we've already dealt with, two chemicals that we've spent quite a bit of time talking about in this class fall into that category. Other than DNA and RNA. What's the chemical we probably spent the most time talking about this whole semester? Comes up just about every day. How about that one? ATP. Same structure. And then the one that we're talking about during cell communication, that was a secondary messenger. What's the name of that chemical? CAMP. All those guys are nucleic acids. Now, before we go any further, let's talk about what makes a nucleic acid be a nucleic acid. Let me go back to a clean screen here. Maybe I won't be able to go back to a clean screen. All right, nucleic acids are made up of, well, first of all, nucleic acids are polymers, which means they're what kind of chemicals? Polymers. What's the word polymer mean? Many. They're made of many pieces, repeating pieces, right? What's each individual piece in a polymer called? Polymers are made of monomers. If we were talking about proteins, proteins are polymers. What are the monomers that make up proteins? Amino acids. If we were talking about starch, starch is a polymer. What are the monomers that make up starch? Glucose, right? If we're talking about nucleic acids, they're polymers. The monomers that make them up are called nucleotides. And you need to know what a nucleotide is. A nucleotide itself is made of three pieces. And you can see those over here. This part that I'm circling, that's one nucleotide. Here's another nucleotide. Nucleotides are made of three things. The P stands for what? Phosphate group. Anytime we see those nice hexagons or pentagons, what are those? Hexagons and pentagons are sugars, for the most part. Carbohydrates, so that's a sugar. In DNA, that sugar is called deoxyribose. Carbohydrates names usually end in three letters. OSE, so it's a sugar. And then the third thing are these guys, the T's, the A's, or the C's, and the G's. And together, as a group, those are called what? Nitrogenous bases. And we'll see why they're called nitrogenous bases in a minute. But one phosphate connected to a sugar, connected to a nitrogenous base, those make up a nucleotide, one nucleotide. And notice what's always in the middle. Sugar. What are those numbers? What are those numbers numbering? The 1, 2, 3, 4, 5. What are those numbering? The carbons. Remember, everywhere there's a bend, there's a carbon. So always attached up to carbon number 1 is what? What is that? Nitrogenous base. Always attached to carbon number 5 is phosphate group. You need to know that. Attached to carbon 1 in the sugar is the nitrogenous base. Attached to carbon 5 is the phosphate. You should know that. And notice down here around carbon 2 and 3, there's not anything. They're sort of open. But attached to number 1 is the nitrogenous base. Attached to number 5 is the phosphate. Now, in the middle of this picture, something else I want you to notice. Here's a nucleotide. Here's another nucleotide. Here's a nucleotide. Nucleotide. Nucleotide, and so on. Phosphate, sugar, base. Those make up nucleotides. Let me go to another version of this picture, so we don't have to look at all that writing. All right, on here. Notice that if we were talking about a ladder, what we might call the backbone of our ladder, this part and this part, in DNA, what makes that up? What makes up the outside part of our ladder? Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Same over here, right? And they just repeat. That's the same in every known living thing. Bacteria, fungus, protist, animal, and even viruses that have DNA. It's the same. Can that be the important part of the DNA? It's the same in every living thing. Exactly the same in every living thing. Can't be. Okay? That part's just there to what? Hold the rest of it together. That's the backbone of the ladder. The important part of the ladder is what? Part step up on. What do we call those steps? What do we call the steps on the ladder? What do you call the steps on the ladder? You guys ought to know this.
Rungs.
They call the rungs, right? The rungs of the DNA, the parts you step on. Those are made up of pairs of nitrogenous bases. And I'm hoping you guys already know that these things have to pair up in a certain way. Guanine always has to pair up with C, and thymine always has to pair up with adenine. Now, what's holding these guys together? Where we see these dots in our chemical pictures, what are those dotted bonds? What does that mean? Hydrogen bonds. What does that mean about A and T and G and C? What kind of chemicals do they have to be if they're making hydrogen bonds with each other? What kind of chemicals make hydrogen bonds? There you go. Somebody said it. What kind of chemicals make hydrogen bonds? Polar. Why are they polar? What element, electronegative element, do they have a lot of in them? Nitrogen. Remember, the electronegative things are in the top right-hand corner of the periodic table, right? Nitrogen, oxygen, fluorine, chlorine. Those are the main ones. So they're electronegative. They form hydrogen bonds. Now here's the thing, and it's kind of scary to think about this. What do we know about hydrogen bonds? They're really weak. They break easy. So your DNA is held together by bonds that break really easily. And at first, that might make you a little uncomfortable. But it turns out we need them to break. But 2 times during the life of the cell do they need to break. Well, yeah. Let's be a little more specific. Whenever the DNA needs to what? Replicate, copy itself. Those bonds have to break. When else? When the DNA needs to be used to make what? What's DNA used to make? What's DNA's job? To code for proteins. But before it can make proteins, it has to make what? DNA can't get out of the nucleus. It has to make RNA. And what do we call that process when DNA is used to make RNA? Transcription. So those bonds have to break during replication and during transcription. If they were strong bonds, that couldn't happen. So those hydrogen bonds are important. We want them to break. Let me back up a few slides. Now, how long have we known about DNA and the way it works? When did we discover the structure and function of DNA? Somebody give me A guesstimate as to when that might have happened. Way past that. 1950s. About 1953. So in terms of history, 1953 is not that long ago. It seems like a long time ago to you, but it's not that long ago. 1953 is when we figured out DNA. Before then, before the 1950s, what chemical do you think scientists thought was the important genetic material? They didn't think DNA was important before the late 40s. What do you think they thought was the genetic material? Let me back up a step. When was Mendel around? With the pea plants and the Punnett squares and that kind of stuff. Late 1800s, 1880s, 1890s. So they knew there was some genetic stuff going on back in the 1800s, but they didn't know what chemical was responsible for it. What chemical do you think they thought was responsible for genetic information? Protein. And they're sort of right. They're just what? You just missed a step, right? Because DNA leads to what? Proteins. Why do you think they thought proteins?
Because they're sort of like the way the DNA transfers protein to protein is a little bit different.
Yeah, well, and two, proteins are these big, complicated, twisted molecules, right? Got 3 or 4 different levels of structure. They thought genetics had to be really complicated. DNA is kind of a simple chemical. They thought it had to be something complicated for this to work. So for years they thought protein is the genetic material. But then in the 40s, some people started doing some experiments. That led them to think that, well, maybe this DNA stuff is important. The first guy was a guy named Griffith, Frederick Griffith. I'm going to show you what he did here first. He did what's called a transformation. Before we even talk about what that is, what does transformation mean? Change. I'll just go ahead and define transformation for you right now. In terms of genetics, a transformation is when an organism takes in DNA from something else. And what? If you take in DNA from something else, what's going to happen to you? What's the name of the process, right? You're going to transform. You're going to change because of that DNA. Now, here's the thing with Griffith. Griffith was working with 2 different kinds of pneumonia bacteria, pneumococcus bacteria. One was called the S strain, one was called the R strain. The S strain bacteria had a capsule on the outside, like a mucus coating basically around it. The R strain didn't. Otherwise, they were pretty much the same. What would a capsule, a mucus coating on the outside, what might it help a bacteria do? Might protect it from drying out, but in this case that wasn't that important. What else might it protect it from? Immune systems. So here's what Griffith did. He took the S strain bacteria, the one with the coat, injected it into mice, and the mice died. He took the R strain, which was the same but didn't have the capsule, injected that into the mice, and they were fine. They didn't even get good and sick. That's kind of what he expected. Then he took the S strain and he killed those with heat. Injected those into the mice. They lived. Well, that made sense. Should dead bacteria be able to make you sick? Nope. Here's the part that was confusing. He took the heat-killed S strain, these guys, and the live R strain, these guys, neither one of which made the mice sick, and injected them at the same time. And when he did, notice what happened. The mice died. Griffith didn't know what caused that. He said that these R strain bacteria were taking something from the S strain bacteria, the dead ones, and changing. But he didn't know what that thing was. He had no idea. So a few years later, another guy comes along, Avery, and he does the same experiment. But he's able to figure out what were these guys taking in from the dead S strain. What were the living R strain taking up from the dead S strain? What was still floating around that they sucked in it? DNA. They took in plasmids, little pieces of DNA. And that changed them. So this was the first experiment that showed people that, hey, this DNA stuff, it might be important. Because it can change what something is. It can change a bacteria from one kind of bacteria to another. So this was the first experiment that let people know, hey, maybe DNA is the genetic information instead of proteins. Some other guys came along, and they were working with bacteriophages. What are those? Bacteriophages. They're viruses that attack bacteria, right? In this picture, this is a real picture. This is the bacteria. What kind of bacteria is that, since it's shaped like a capsule? What's the name for rod-shaped bacteria? Bacillus. Yep. Bacilli or bacillus. So that bacillus is getting attacked by lots and lots of viruses. Each one of these little things is a bacteriophage. And notice it's getting attacked by many, many, many of them. Now let's think back to the parts of a virus. A virus turned out to be a good thing to use in this experiment. Because what were the two competing things that the scientists thought might be the genetic material? DNA and protein. Well, what's a virus made out of? DNA and protein. So here's what they did. They first of all, let's review. All the stuff in yellow up here, that's protein. The yellow part of a virus, the outer coat, is called what? The capsid. And then inside the capsid is the DNA. Or in some viruses, it's what? All right, they're working with DNA viruses, so it doesn't matter here. DNA virus. Now we know that typically these viruses attach to what? How does a virus get in? How does it get its DNA into your cell? What does it attach to on your cell? It usually attaches to a receptor for something else, and it uses it like a doorway to get its DNA in. So that's what was going on here. Now what was really cool about the experiment here from these two guys, Hershey and Chase, by the way, the two guys doing this. They took the virus and the bacteria. They grew them in a medium in food that had radioactive phosphorus in there. Radioactive phosphorus. Does protein have phosphorus in it? Is there any protein in phosphorus? Is there any phosphorus in protein? No. Is there phosphorus in DNA? Yes. So basically what they did is they made the DNA of the virus radioactive. But not what part radioactive? Protein part. The capsid didn't become radioactive. So then they put the virus and the bacteria together. Let them grow together. After a while, they tested for where all the radioactive stuff was, using like a Geiger counter. Where did all the radiation end up? Inside or outside the bacteria? Inside. So that was able to show that what the virus was injecting into the bacteria was what? DNA. But just to make sure, they did a little slightly different version. This time they grew the virus in radioactive sulfur. Is there any sulfur in DNA? No. But there is sulfur in what? Protein. So this time they made the protein part be radioactive. And they tested for where the radiation was at. And where was all the radiation? Outside, not inside. So after Avery and Griffith and Hershey and Chase, basically there was very little doubt that DNA was the stuff. DNA was the genetic material. So now there became a race because this was huge. Everybody knew now that whoever figured out what DNA is shaped like and how it works, they're going to be famous. Because they're going to be in every textbook ever written from now on, every biology textbook ever written. So there was sort of a race to figure out how it was shaped and how it worked. Now back in the day, there was a really famous scientist. His name was Linus Pauling. And a lot of people thought he would be the guy who figured this out. Anybody ever heard of Linus Pauling before?
No, Linus.
Different Linus. He's one of the few scientists who have won 2 Nobel Prizes. He won one of them because he figured out how covalent bonds work. That's pretty important, figured out how covalent bonds. The other one, he sort of turned into a hippie, and he won a Peace Prize. Anybody know what he was protesting against? What were the hippies protesting against? Well, Vietnam and? Well, that too. Nuclear weapons. Okay, he's an anti-nuclear weapon guy. He won a Peace Prize for that. A lot of people thought that he was the leading candidate to figure this out. Turns out that he had a son who was friends with these two guys, Watson and who? What's the other guy's name? Watson and Watson and Crick. Crick, like cricket without the et. Now there are some people who think that Pauling's son was feeding information to his two friends, that his father has doing experiments and that he was sneaking information to his two buddies. Not sure about that. But we do know that Watson and Crick did no experiments. They weren't your typical science. They did 0 experiments. Basically what they did is they took information that other people had gotten from experiments and used it to build a model of how they thought DNA would work. They basically came up with a puzzle, and they started putting the puzzle pieces together based on information they got from other people, and eventually they figured out how the pieces fit together. So they figured out the shape of DNA without doing any experiments. They did that in 1953. And a lot of people would argue that Watson and Crick's finding out the structure of DNA and how it works in 1953, a lot of people would argue that that's the most important scientific discovery ever made. Because eventually, what's it going to lead to? What are they going to be able to do with genetics one day? Soon. If you have, if you need a heart, they're going to be able to genetically make you a heart, and it'll be your heart. I mean, they already. Let's say you have, you've had this dog for 10 years, and your dog, you let it out of the fence today, and it runs out the road and gets hit by a car, and you love that dog, you want that dog. You can actually collect a blood sample from that dog, mail it to a company, and they will clone that dog and send you an identical copy of that dog. Okay? Now here's the thing. Yeah, genetically, it's an identical twin of your dog. Personality-wise, it may be very different. Okay? It's not going to have any memories of you. Okay? Genetically, it's the same dog, but memory-wise, it's not. I mean, it's just like two identical twins.
Like grown up?
No, it would be a puzzle. No, but it's like two identical twins. They can be pretty different.
You know how much it is? It's like $200,000.
It's really expensive. Yeah. Go ahead.
Hey,
listen, guys.
What's this topic here? This guy's talking about how this one man had discovered how to create square-on skin. It's a little purplish fluid, right?
Guys, he's trying to talk here.
And basically, if you have like a really bad burn, like 3rd-degree burns, and you like have no skin on a certain part of your body, they can just spray it on there. Put a type of gauze or some sort of wrap on it.
And it'll grow and cover the hole. Yeah. There's all kinds of cool stuff that this is going to lead to. If you have certain kind of diseases, eventually they're probably going to be able to go in and put different genes in you to fix those diseases. They're getting there. So this is going to be huge. And we're talking about stuff that's going to happen in your lifetime, soon. So, huge discovery. Watson and Crick. Now, turns out Watson's a jerk. Watson's still alive. He's 80-something years old now. Right after this discovery, he wrote a book called The Double Helix. And you might have heard of that book before. And in that book, he does a lot of bad-mouthing of what group of people? Women. He's a very anti-woman guy, sexist guy. No, not gay. He's opposite of that. Okay? He's just very anti-woman. He thinks women ought to be in the house cooking dinner and right? And he doesn't mind saying that. He's also made some pretty racist comments against black people over the years. Okay? He's not a good guy. Okay?
What does he talk about in his book?
He basically, one of his ideas, just pretty recently, was how genetically superior white people were to black people. That was one of the comments that he made. Okay. No, I'm not saying it's true. He said. He's a very smart guy, but he's one of these guys who is very smart but has no idea socially how to act.
Dr. Dean.
Yeah.
I want to write the book on Watson. You're just Watson. I believe my fist is more genetically superior than your face.
Yeah. But Watson, he was in town just a year or two ago in Huntsville, speaking at somewhere out on Redstone, I think. And he's still a famous guy. He was part of the Human Genome Project. You guys know what that is?
Yeah.
Where they figured out what each gene in the human body does. He was part of that until eventually he made some comments that he shouldn't have made, and he got fired, basically, from that particular project.
You just say so you can say.
Well, I'm Crick.
No.
But let's talk about Crick died. Crick was older. Crick's been dead for a lot of years. Watson, when they figured this out, Watson was in his 20s. And Crick was, I think, around 40. So Crick's been dead for a while. Here's the deal, though. Another place where they did somebody wrong, sort of. Back in 1951, a woman scientist, Rosalind Franklin, took what's called an X-ray crystallography picture of DNA. Watson and Crick knew her, but they didn't like each other. So she didn't like Watson very much. They ended up, though, getting her picture through a backdoor source. They didn't get it from her. And they used her picture without her knowing. And that was one of the key pieces to them figuring out how all this worked. And guess what? They never did. They never gave her any credit for it. She ended up getting cancer, probably from using all the x-rays, and died. They got a Nobel Prize. She got nothing. Okay? Another guy whose work they used was a guy named Edwin Chargaff. Now, I'm not bad-mouthing them for using other people's stuff, because that's what scientists are supposed to do, right? They're supposed to read everything and use what other people have found. But typically you use it with their permission, right? They didn't always do that.
Give them credit.
And you give them credit. All right. Chargaff, though, here's what Chargaff did. Well, first of all, here's Rosalind Franklin's picture. I don't know exactly how to read this picture, but from this picture, they were able to tell that DNA had two strands and that it was curved. It was helical. I don't know how to read that from that picture, but they were eventually able to do that. Here's what Chargaff did. Chargaff tested all kinds of different living things. People, different kinds of bacteria. These are fruit flies. All kinds of different things. And the pattern that he saw was that the amount of adenine was pretty much always exactly the same as what? What's A almost the same as every time? Thymine. That in every species, these two seem to be paired up. And that the amount of guanine is almost always the same as the amount of cytosine. Not exactly, but really close. Chargaff didn't really know what that meant, but he published it. Watson and Crick found it, read it, used it, and eventually, by building this model over and over again, they were able to figure out how it all went together. They put Franklin's work, Chargaff's work, Pauling's work all together, and they found a model that worked for DNA. And once they figured out the model, the shape, the structure was pretty obvious. The function, rather, was pretty obvious in terms of how it worked. So here's what they came up with. First of all, we already said that DNA is made out of nucleotides. DNA is a polymer. Nucleotides are the what? Monomers. Each nucleotide we know is made out of sugar in the middle. And that sugar in DNA, deoxyribose. It's a 5-carbon sugar.
Is this all?
Attached to that 5-carbon sugar on one side, the 5-carbon side is the phosphate. It's attached up to carbon number 5. And attached up to carbon number 1 is the nitrogenous base. Now what they were eventually able to show is that A and T pair up in the middle of the double helix, and G and C pair up in the middle of the double helix. We sometimes say they're complementary. Now, in a minute, we're going to watch a video that talks about these two different kinds of nitrogenous bases, purines and pyrimidines. Two different groups of nitrogenous bases. Notice that A and G are purines, and C and T are pyrimidines. Now let me show you what that means. These are the purines. Notice here's a sugar, a phosphate, and up here, that's the nitrogenous base. Sugar, phosphate, nitrogenous base. Maybe you won't pay attention to, but notice those two nitrogenous bases are pretty big. They're big rings of carbon and nitrogen. They look like two rings fused together. Those are the purines. The pyrimidines, these guys, sugar, phosphate, nitrogenous base. Sugar, phosphate, nitrogenous base. Notice these are smaller. They're only one ring. But each group, either A and T or G and C, is made of one of these, one pyrimidine, connected to one purine. One big nitrogenous base connected to one small nitrogenous base. That's always the case. Purine and pyrimidine together make a pair. Now, here's the basics of the Watson and Crick model. They were first able to show that DNA is made of two chains of nucleotides. It's a double helix, two chains. And that those two chains are sort of like a ladder that's been twisted into a helix shape. So two chains of nucleotides twisted into a 3D double helix shape. We already talked about how that the backbone of the DNA is made of phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Now this part we haven't talked about, that the two strands are anti-parallel. Let's go to a picture and talk about that for a second. Notice how the point of the sugar down here is pointing down, and the point of the sugar here is pointing up. The sugars are in opposite directions. This piece of DNA is running this way, and this piece is running that way. Parallel to each other, but what? But in the opposite direction. Therefore, it's called anti-parallel. The two chains are reversed. One's running one way, one is running the other. Sometimes this end down here with the point is called the 5' end of the DNA, because the outermost part of the sugar is this 5 carbon sticking out down here. Up here, the flat end of the sugar, that's where the 3 carbon is at. So sometimes we call this the 3' end. On the other chain, this is not going to be a 3 end, this is going to be what? This is going to be a 5 end, and this is going to be a 3 end. So they're opposites. The ends are opposites. They're anti-parallel ends. You'll talk more about that in the video here in just a second. No? The 3' end is just this flat part of the sugar. The 5' end is the more pointed part of the sugar. So down here's the point, so that's 5' end. Up here's the flat part, that's 3' end. All right, the other thing that they came up with was the whole complementary base thing, that A goes with T, G goes with C. Those form the rungs in the middle of the ladder, and they're held together by hydrogen bonds, because nitrogenous bases are polar kinds of molecules. Now, we're going to look at about 2 more slides, and then we're going to look at a video. Number 1, DNA. What's its main and really its only job? Make proteins. And then what do proteins determine about you? Everything, your traits. Proteins determine your traits. DNA codes for proteins. Proteins determine your traits. They determine who you are, what you like. By coding for proteins, DNA does all this other stuff. They store your genetic information. DNA can self-replicate or self-duplicate. It's passed on and inherited. It expresses the genetic message. What is the genetic message? Make what? Make proteins, determine traits. And as we were saying before, the backbone of DNA is phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Can that be the important part of the DNA? If it's the same and exactly the same in every living thing, can it be the important part? No. The important part is what part? The rungs, the nitrogenous bases, and they're like a code. How many nitrogen-based letters does it take to make up one code word in DNA? How many A's, T's, G's, and C's make up one code word in the language of DNA? 3. combinations of 3 nitrogenous bases code for one way. Every 3 nitrogenous bases code for one way. What are we trying to code for? Yeah, but what are proteins made out of? What do we got to put in order to make a protein? Amino acids. Every 3 nitrogenous bases code for one amino acid. And a lot of amino acids put together in the right order give you what? A protein. So what are those three-letter groupings of nitrogenous bases called? What are the 3-letter words in the language of DNA called? The code for one amino acid. They're called codons. Every group of 3 nitrogenous bases codes for one amino acid. They're called codons. It takes a lot of codons to make a protein because proteins are big, long, complicated chemicals. All right, one other thing that I want to talk about before we look at the video is this slide. Every living thing has DNA. But we want to talk about differences between prokaryotic and eukaryotic DNA, in particular the chromosomes. The way DNA works is very similar. It codes for proteins in bacteria, it codes for proteins and everything else. But the shape of the chromosomes is different. In bacteria, typically the chromosomes are circular shaped. They're loops. And usually how many of them are there in a bacterial cell? Just one. Now usually there's also some extra little bitty pieces of DNA. What are those little bitty pieces called? Plasmids. And what are those mostly used for? Plasmids. What do they do with plasmids? They conjugate and they do what with them? Swap them. That's kind of like bacteria sex, right? It gives them some variation. But in terms of their chromosomes, they're really small. Does a bacteria, since it's pretty simple, need a whole lot of genes and a whole lot of proteins? Not really. So it doesn't need a whole lot of DNA. There are a few enzymes and proteins attached to that DNA, but not very many. And also we know that prokaryotic DNA is not in a nucleus. It's just sort of floating around. What do we call the bacterial DNA? It's not a nucleus, it's what? Nucleoid. Nucleoid. It's called the nucleoid. And also bacteria are missing the proteins that our cells use to wrap up the DNA. What do we call those proteins that our DNA wraps around to make a chromosome? Histones. So bacteria don't have those. No histones. With eukaryotic DNA, our DNA, our chromosomes are said to be linear. Now don't think they're straight lines. They're not. They're just not loops. They're not closed loops. Compared to bacteria, our chromosomes are much longer, much bigger. We have a lot of chromosomes in every cell. Jacob, join us here. How many chromosomes in a typical human cell? How many? 2 sets of 23. So 46, right? 2 sets is important because what do we call our cells since we have 2 sets? Diploid. But there are other things that have more chromosomes than us. Realize though that eukaryotic cells usually have several chromosomes in every cell, and that we have these histones. Histones act like spools. Our DNA winds around those to make the chromosomes and to organize itself. Also note that our DNA is found in a nucleus, enclosed by what? Nuclear membrane, or what's the other word for it? Envelope, nuclear envelope. The whole thing about a nucleosome we'll talk about later. For right now, here's what you need to know. They both have DNA, prokaryotes and eukaryotes. Chromosomes in prokaryotes tend to be circular, and eukaryotes tend to be linear. In prokaryotes, those chromosomes tend to be really, really small. A whole lot bigger in eukaryotes. In prokaryotes, it tends to be 1 chromosome. In eukaryotes, it tends to be a lot. Eukaryotic chromosomes tend to have DNA spooled around histones. Prokaryotes, they don't have those. And also the eukaryotic DNA is enclosed by a nuclear membrane, a true nucleus, not true in a prokaryote. And that's where we're going to stop. with this for right now.